Good afternoon, everyone. On behalf of the Center for Brain Health, welcome to Frontiers of Brain Health, where we take a deep dive into the most exciting new developments in brain research. I'm Francesca Philby. I'm a professor and researcher at the Center for Brain Health, where I direct the Cognitive Neuroscience Laboratory of Addictive Disorders. For those of you who don't know much about us, the Center for Brain Health is a cognitive neuroscience research center at the University of Texas at Dallas. We have dedicated the past 3 decades to exploring neuroplasticity and the brain's amazing lifelong potential to get stronger and work better. Stay until the end to hear about our groundbreaking research study, the Brain Health Project. Today, I have the pleasure and privilege of introducing one of the world's leading addiction scientists, Dr. Eric Nestler. Dr. Nestler is one of the most comprehensive scientists that I truly know, as he applies the bench-to-bedside approach to research. He is a trained psychiatrist that utilizes animal models to understand the underlying mechanisms of disease to improve treatment. Dr. Eric Nestler is the Nash Family Professor of Neuroscience, Director of the Friedman Brain Institute, and Dean for Academic and Scientific Affairs at the Icahn School of Medicine at Mount Sinai. His research topics include addiction, behavioral health, depression, epigenetics, molecular biology, neuroscience, stress, and many, many more. He has published more than 650 peer-reviewed articles and is also the principal investigator on 6 NIH grants. So as you guys can see, he's a very busy person, and we are so very lucky to have him this afternoon. So thank you, Dr. Nestler, for your generosity. To the audience, please note that we will have a few minutes at the end to answer a few questions. So to do so, please use the Q&A function, and we'll get to as many of your questions as we can at the end. Also, please be aware that this presentation is being recorded. And so without further ado, my friend and dear colleague, Dr. Eric Nestler.
Francesca, thank you so much for the opportunity to speak here today. I only wish I could be in Dallas in person. I had such a great 8 years in Dallas at UT Southwestern, and it's great to be back, at least virtually. So I'm going to share my screen and get started with my talk today. As Francesca mentioned, I'll focus on our work on the transcriptional and epigenetic mechanisms of drug addiction. Let me begin by putting this in some context. We know that all drugs of abuse interact initially with proteins located at the synapse, such as neurotransmitter receptors or transporters or channels, but that addiction requires repeated exposure to a drug of abuse. And we and many other groups over the years have suggested that because of the long-lasting nature of the behavioral abnormalities that define addiction, that alterations in gene expression are involved. That addiction can be viewed as a type of drug-induced neuroplasticity mediated at the level of gene expression. So the way that would occur is that drugs of abuse, by repeatedly perturbing these extracellular synaptic mechanisms, would trigger changes inside of nerve cells and eventually signal to the nucleus, where drug-induced changes in proteins called transcription factors, which bind to DNA in a sequence-specific manner, would alter the activity of specific genes in nerve cells and thereby mediate the stable changes in neural and circuit function that underlie the behavioral abnormalities. And let me just state at the outset that all current treatments for addiction focused on this limited view of the initial extracellular actions of drugs of abuse, leaving unexplored literally thousands of proteins at the intracellular level. And I'll come back to this at the end of my talk. In order to identify the changes in gene expression that contribute to drug of abuse, it's necessary to know where in the brain to look for the changes. And for that, we've relied on decades of research to identify what are called so-called brain reward regions, areas of the brain where drugs of abuse produce their initial rewarding effects and presumably perturb the systems, the circuits, to cause behavioral abnormalities that underlie addiction. The best characterized brain reward regions arise in dopamine neurons in the midbrain and their projections to the forebrain, in particular to the nucleus accumbens, where I'll be focusing today, although I want to emphasize the importance of many other limbic structures like amygdala, hippocampus, frontal cortex, and others in mediating the lasting actions of drugs of abuse on the brain. Now, historically, my lab focused on two types of transcription factors, termed delta phosB and kreb. We focused on delta phosB initially because it was the only member of the phosgene family of transcription factors, referred to as AP1, that is induced after chronic drug exposure. And delta phosB was interesting because it showed a very unique temporal pattern. That's shown in the middle cartoon, where an acute drug exposure would rapidly induce C-phos and other phos proteins, and only to a slight extent induce delta phosB. But because, unlike all these other phos proteins, which are very unstable, you see they degrade rapidly, delta phosB is very stable, which means that this small amount of delta phosB protein induced with each drug exposure accumulates during a course of repeated drug exposure to the point where delta phosB becomes by far the predominant phos family protein present in these nerve cells, and this is focusing, as I mentioned, on Nucleus accumbens. The other protein that we've looked at is kreb, a transcription factor that mediates the effect of the cyclic AMP pathway on gene expression, and we focused on kreb because of earlier data implicating the cyclic AMP pathway in various aspects of drug action. The temporal properties of kreb are much more typical. You see this spike and trough pattern with acute drug exposure as well as repeat drug exposure. And then over the years, including my 8 years in Dallas, we provided a great deal of evidence for the functional consequences of delta phosB and kreb induction in the nucleus accumbens in the context of drug exposure. First, we know that delta phosB is induced only in one type of nucleus accumbens neuron. The projection neurons of nucleus accumbens come in two types, those that predominantly express the D1 dopamine receptor, the other one predominantly expressing the D2 dopamine receptor. Delta phosB is induced by most drugs solely in D1 type neurons, where when it's induced, it increases an animal's sensitivity to drug and natural rewards, leading to a positive emotional, motivational state. And with David Self and colleagues in Dallas, we showed that this drives drug craving and relapse, perhaps through positive reinforcement. Kreb in many ways can be seen as doing the opposite, as serving a classic negative feedback homeostatic mechanism of drug tolerance and dependence. First of all, kreb is activated in both D1 and D2 type medium spiny neurons. It has the opposite effect of delta phosB, where it reduces responses to drug and natural rewards, producing a negative emotional state. And again, with David showing that this also drives self-administration, drug craving and relapse, although through a process of negative reinforcement. Now, in more recent years, we've tried to move beyond transcription factors per se to mechanisms of what we call epigenetics, which is simply referring to chromatin mechanisms. The word chromatin refers to the material inside of a cell nucleus, where we know that the DNA double helix in a mammalian organism comprised of 3 billion bases. Each one of these lines, steps on a ladder, would be considered a base. 3 billion bases of DNA stretched out linearly would be about 2 meters in length, and that fits within a microscopic cell nucleus. And over the past 2 decades, we've learned a great deal about that compaction. We know that the DNA double helix is wrapped around octamers of histone proteins to form the unit of chromatin called a nucleosome. Nucleosomes are then further organized in very rote ways in their most condensed form in what we would recognize as a chromosome. This organization becomes functionally important because DNA in these spans where nucleosomes are tightly packed are inactive. They can't serve as a template for RNA transcription, whereas DNA in more loosely spaced areas of chromatin can be functionally active. This means that we could use chromatin information, so-called epigenetics, to help identify drug-regulated genes, to understand transcriptional mechanisms in vivo, And by analogy to developmental biology and cancer biology, where certain types of chemical modifications open and close nucleosomes, once they occur, are permanent, the hypothesis here is that drug exposure would also produce these near permanent changes in chromatin structure, turning off or turning on genes in a persistent manner. Now looking at this level of analysis in greater detail, and this is the most complicated slide that I'll show you, so please bear with me, is now the DNA are these gold stripes, the DNA double helix wrapped around the octamers of histone proteins represented by these spheres, showing you the two opposite ends of the spectrum of chromatin, open or active chromatin, where nucleosomes are spaced apart, and condensed inactive chromatin, where nucleosomes are tightly packed together. These are dynamic transitions between the two states mediated by hundreds of proteins. So for example, transcription factors, as I mentioned, we've been interested in delta phosphy and kreb, would bind to relatively open regions of DNA where these proteins can gain access to the DNA, and then recruit co-activators. A co-activator might include a histone acetyltransferase that adds an acetyl group to histones to move these nucleosomes further apart, and so on, leading ultimately to the recruitment of the RNA-2 polymerase basal transcription complex to mediate gene transcription. At the opposite end of the spectrum, condensation of chromatin is also a highly active process involving hundreds of repressor proteins. For example, an HDAC, a histone deacetylase, would remove these acetyl groups and allow these nucleosomes to become more tightly spaced together. So in recent years, since we moved to New York, now about 13 years ago, we decided to take an open-ended approach based on the idea that we don't yet know enough about addiction to always guess what the right mechanisms to focus on might be. And so, for example, we would start out using next generation sequencing technologies like RNA-sequencing, which would enable us to identify an entire subset, all the RNAs that are induced within a particular brain region, for example, Nucleus accumbens, in response to chronic exposure to cocaine or another drug of abuse. Another set of RNAs would be downregulated. If these RNAs are regulated transcriptionally, then those changes would have to be associated either with an increase in a chromatin mechanism associated with gene activation. For example, I mentioned histone acetylation, or a reduction in some repressive chromatin mechanism. And we would measure these chromatin mechanisms genome-wide using a related approach called CHIP sequencing, chromatin immunoprecipitation followed by sequencing. We would also be able to use CHIP sequencing or related approaches to map exactly where it is that kreb, delta phosphy, and other transcription factors are binding to the genome. Together, this is an enormous undertaking involving 20 or so terabytes of sequencing information for a single brain region, single time point, just for the measures shown in this slide, but providing an invaluable, open-ended, unbiased data set at which regions of the genome are being affected by cocaine action. So let me illustrate how we would use these data sets. This is an experiment published a couple of years ago by Dina Walker, who's now in Oregon, and Erin Kalapari, who's now at Vanderbilt. And what they did was to have mice self-administer cocaine. They would press a lever, either get saline as a control or get cocaine repeatedly. We would harvest brain areas from a group of animals 24 hours after the last self-administration session. Other animals were housed normally for a month, at which point they were given a challenge dose of either cocaine or saline. This 30-day withdrawal time point is important because what we call incubation, or the best that we can infer, craving or relapse, builds over time of withdrawal. This is illustrated in the data on the inset. Whereas if we took mice one day after cocaine self-administration, put them back in the self-administration chamber, but turned the cocaine lever off and just asked to what extent do the animals go back and press that cocaine lever anyway, you can see the mice do press the lever. But after 30 days of withdrawal, that behavior is increased. This is interpreted as incubation of cocaine craving or relapse. And at these, under these 6 conditions, then we harvested 6 brain regions among the ones I've already mentioned for RNA sequencing. So a very, very large RNA sequencing data set. I'm just going to show you data for one brain region, the nucleus accumbens. And what we used is machine learning to identify genes that are uniquely regulated at the 30-day time point. So these are animals 24 hours after the last cocaine self-administration session. These are animals that self-administered saline and were just given a single dose of cocaine. Essentially, they're the animal's first acute exposure to cocaine. Animals 30 days after cocaine self-administration that received a saline challenge, and animals that self-administered cocaine after 30 days withdrawal then received a cocaine challenge. And you can see dramatic reprogramming of the nucleus accumbens transcriptome in these heat maps. The heat maps show genes that are upregulated in yellow, downregulated in blue. Each of these vertical lines that you could see represents a single RNA that's induced or repressed. And here are genes that are up or down regulated uniquely at the 30-day time point, regardless of whether the animals got a challenge dose of saline or cocaine. And on the right, the genes that are uniquely regulated in response to that priming dose of cocaine. Let me point out one additional feature of this graph at the right, where you can see that the second heat map that I'm pointing to with my cursor is acute cocaine. And the last heat map is the same acute cocaine, but in animals with a very different month-ago history of cocaine self-administration. Look at how many genes are primed for greater induction or desensitized for less induction for repression and expression in response to a distant history of cocaine self-administration. Now, one can go further and relate these changes in gene expression to behavior. So now what I'm showing you in this heat map, these horizontal lines, yellow and blue, don't represent genes, they represent behaviors. All the features of self-administration behavior that are seen in these animals, then using machine learning to create what we call an addiction index. Obviously, to talk about something like addiction in a mouse is a big inference. Nevertheless, we believe that animals with a higher addiction index show more addiction-like behavior across composite behavioral scores. So you can see animals that self-administered saline all had low addiction index ratings, as you'd expect. But among the animals that all learned to self-administer cocaine, a wide variation in addiction-like behavior, even though these animals are all genetically identical. We could come back to that during the question period if you're interested. And then what we did was to relate the gene expression to the addiction index findings. So now what I'm showing you are all the genes that are upregulated in yellow, downregulated in blue, positively associated with the addiction index in red, negatively associated in gray in nucleus accumbens. And you can see some interesting patterns. Note that in response to acute cocaine exposure versus in response to that priming dose of cocaine in animals with a history of cocaine self-administration, there are opposite patterns of gene expression. Genes that tend to be downregulated acutely are induced chronically, for example. This is interesting because it suggests that an animal's initial transcriptomic response in the nucleus accumbens to the first ever exposure to cocaine is actually predictive of whether the degree to which that animal will go on to get addicted to cocaine, as we can infer in a mouse model, even though the direction of regulation might be quite different. This doesn't hold for any of the other brain regions studied, for example, hippocampus as a control. Then we can go into these data sets and ask, of everything that's known about the genes that are up or down regulated in association with the addiction index, what are the predicted transcription factors that one would expect to be driving those changes? This is the list that we obtained, and note to our we were very happy to see that kreb and AP1, meaning delta phosphB, were highly implicated as driving these transcriptional changes. Sometimes, one can guess correctly even in the absence of truly open-ended data. Nevertheless, we still identified many additional types of transcription factors not previously implicated in drug exposure. My lab is now studying these other factors. Now, everything that I've talked about thus far has relied upon RNA sequencing data on bulk dissections of nucleus accumbens tissue, even though, as I mentioned at the outset, that the nucleus accumbens has made two major subtypes of neurons, 95% of which are called medium spiny neurons that come in two subtypes, those that express D1 receptors or D2 dopamine receptors. In general, the D1 receptors overlay with the direct projection pathway of striatum. These D1 expressing nerve cells project primarily back to the midbrain, although a small subset projects elsewhere, whereas the D2 expressing neurons overlay onto the indirect projecting pathway to the ventral pallidum. And this is important because these two nerve cells play very different roles in drug addiction. And I'm just highlighting here two papers from my own lab, Mary Kay Lobo at Maryland, Aaron Calipari I mentioned at Vanderbilt. Many other labs contributed to the conclusion stated in this slide. First, that when one controls the activity of these two types of nerve cells experimentally, one sees opposite effects on drug reward. Activation of D1 medium spiny neurons of nucleus accumbens promotes drug reward. Activation of D2 neurons has the opposite effect. Likewise, drug exposure has opposite effects on the activity of these cells in awake behaving animals. So acute exposure to cocaine tends to activate D1 neurons and suppresses the activity of D2 neurons. Chronic exposure exaggerates those differences still further. So it became critical to gain greater granularity into gene expression changes on a cell type specific basis. This is work done by Phil Muse in the lab, and what I'm showing you is another epigenetic assay that's available called Atax sequencing, which essentially provides a measure of the degree to which nucleosomes are spread apart or close together. A higher peak means that the nucleosomes are spread apart. They're open regions or active regions of chromatin. I'm showing you graphs that are focused over what's called the transcription start site of genes. This is at the beginning of the promoter of genes, average genome wide. First of all, when one does this analysis separately on D1 and D2 medium spiny neurons, note under control conditions, the pink graph, that D2 neurons tend to be much more open, about twice as much as D1 neurons. In fact, this correlates with our knowledge that D2 neurons express many more genes at baseline than D1 neurons. Their chromatin tends to be much more open. But notice the dramatically different effects of cocaine. Acute cocaine causes a striking opening of nucleosomes in D1 medium spiny neurons, sustained after 30 days of withdrawal. This is a very long-lasting effects of chronic cocaine administration, and with a priming dose of cocaine opening that chromatin still further. Notice how these changes are much more blunted in D2 medium spiny neurons. Let me emphasize, these are genome-wide averages. So there are examples of genes that are opening and closing in both cell types, but this shows you the composite average across all genes. We also have generated RNA sequencing data sets selectively on D1 and D2 medium spiny neurons, where we've been able to demonstrate dramatically greater gene priming in D1 medium spiny neurons compared to D2 medium spiny neurons. That's illustrated by the preponderance of these yellow induced genes in response to this priming dose of cocaine. And notice the virtual absence of that response of the same genes in D2 medium spiny neurons. Once again, we can go in and infer what transcription factors are important in mediating these effects. And we were delighted to see that the third topmost ranked prime gene is phosphy itself, an effect seen uniquely in D1 medium spiny neurons, again, indicating that sometimes one guesses correctly with non-unbiased data. Now, one could go still further into single cell sequencing results. So this is now, in this experiment, we obtained a population of tens of thousands of D1 and D2 medium spiny neurons. Now we're taking all cells from Nucleus accumbens and sequencing each individual nucleus. One can see when you use single cell sequencing that you can delineate the different cell types that are known to occur in Nucleus accumbens. But some interesting surprises are obtained. This green population of nerve cells, or medium spiny nerve cells, notice how the computer does not easily differentiate those nerve cells that preferentially express D1 receptors or D2 receptors that are distributed rather broadly and not with a sharp boundary across this large population of nerve cells. One can go still further into the details of this and do single cell sequencing on an isolated population of D1 nerve cells themselves, basically taking the positive signal in this panel and further identifying clusters. Well, one can identify over 20 clusters of D1 medium spiny neurons, including a small cluster that also expresses D2 receptors. This overlap is known to occur in less than 10% of nerve cells. Another cluster where delta phosphB is induced, and so on. But I think that this complexity underscores that single cell sequencing is ideal to identify cell types or subtypes of cells, but is not yet ready for prime time, in my view, for looking at cocaine-induced changes in transcript levels. And that's something we can come back to again during the question period if you're interested. So what is driving these long-lasting gene priming effects, this dramatic opening of the chromatin, particularly in D1 versus D2 medium spiny neurons? I mentioned a while ago in my talk that there are chromatin modifications. I mentioned histone acetylation as one example that drive the opening and closing of histones. But there are literally hundreds of types of histone modifications. How do we know which ones to examine? The earlier work we did, which began when my lab was still in Dallas and accelerated when we moved to New York, simply focused on histone modifications that have been found in other tissues to be important. Yet that wasn't particularly gratifying to us because the ability to predict RNA expression changes based on those histone modifications of historic significance were never that impressive. So a few years ago, we decided to use, again, open-ended data sets using proteomics. We collaborated with Ben Garcia and his colleagues at Penn. More recently, one of those colleagues, Simone Sidoli, set up his own lab at Albert Einstein in New York, and we've been collaborating with both to obtain a truly open-ended view of histone modifications as well through proteomics. And what I'm showing you here are changes in different histone modifications. This is just a subset of the data, showing changes that are induced or changes that are decreased in blue under the different treatment conditions that I've already mentioned: acute cocaine, chronic cocaine followed by withdrawal with a saline challenge, chronic cocaine followed by withdrawal with a cocaine challenge. And what I'm highlighting for you in these red rectangles are the most significantly regulated modifications. And by far, the two most dramatically regulated were, let me give you the code here, trimethylation of lysine 79 of histone H3, or what's called H2AZ, which is a variant form of the H2A core subunit of nucleosomes. H2AZ was the most dramatically regulated, including its modification by acetylation, as shown all the way to the right. And we focused on that, and I'll tell you more about that now. We refer to these long-lasting chromatin modifications as chromatin scars. The idea being that a history of chronic cocaine administration is doing something to the chromatin at the vicinity of these regions that is mediating their long-term opening or closing, but that lasts for a very long time. And again, depletion of H2AZ, indicated by the blue, or induction of these H3K79 methylation marks, induction of the dimethylation, depletion of the trimethylation, are marks that we're now looking at. I'll tell you about H2AZ. Philip Muse followed up on these data using chip sequencing for H2AZ and H2AZ acetylation, and consistent with our proteomic data, found profound depletion after chronic cocaine administration. What I'm showing you here in the pink graphs again are control levels, control degrees to which this variant histone subunit H2AZ, or its acetylated form, is present within nucleosomes of the nucleus accumbens. The induction of that incorporation of H2AZ following acute cocaine, but the near virtual complete depletion of the mark after chronic cocaine administration, consistent with the proteomic data set. What was most gratifying about these chip sequencing data is that now when we take the chip sequencing data and look at genomic regions where H2AZ or H2AZ acetylation are depleted, now we can account for a majority of the RNA changes that occur in D1 medium spiny neurons that show that priming behavior. So our hypothesis is that a depletion, the removal of H2AZ from nucleosomes, is what's priming these genes for greater induction. And it's a change that lasts a long time, hence the term chromatin scars. Now, everything that I've told you thus far is correlative in nature. But my lab over the years has devoted a great deal of effort to focus on causality as well. And I wanted to close by giving you one example in which we've demonstrated this type of causality. There's a protein called ANP32E, which mediates the removal of H2AZ from nucleosomes. ANP32E can be seen as a chaperone protein that binds to H2AZ and removes it from nucleosomes. And what we found in our RNA sequencing data, and then later in our proteomic data, is that ANP32E is indeed induced at this prolonged withdrawal time point. So 32 days after chronic cocaine exposure, there's an induction of ANP32E mRNA and protein, and that effect is selective to D1 medium spiny neurons. We also showed, not illustrated in this slide, is that if we knocked out ANP32E, and we did this by overexpressing a microRNA targeting the ANP32E messenger RNA, that we could block the depletion of H2AZ that we showed in this slide here. Consistent with what's been shown in other tissues, that ANP32E mediates this depletion. ANP32E is induced by chronic cocaine, consistent with the depletion. If you knock out ANP32E, you lose the ability of cocaine to produce that depletion. And as an initial read on the behavioral consequences of that action, we found that the knockdown of ANP32E in D1 medium spiny neurons indeed produces prime behavioral effects to cocaine action. This is shown over to the right using a conditioned place preference assay, where animals learn to prefer an environment paired with cocaine exposure, using a higher dose of cocaine that can produce a significant place-conditioned effect under normal conditions, in an animal that received a control viral vector targeting D1 medium spiny neurons in the nucleus accumbens, you see a strong place conditioning effect. But when one knocks down AMP32E, you block this effect, consistent with the ability of AMP32E induction, depletion of H2AZ, mediating this priming behavior. This is now something that we're examining at a much more detailed level. For example, we're studying the effect of AMP32E knockdown in D1 medium spiny neurons on gene priming itself by RNA sequencing, as well as on self-administration and relapse behavior, which will provide a very important extension of the findings shown here. Notice how this ability of AMP32E to control behavioral responses to cocaine is specific for D1 medium spiny neurons, not seen in D2 medium spiny neurons. So this is our model where we would have, after acute cocaine administration, the recruitment of H2AZ and H2AZ acetylation incorporated within nucleosomes, within the nucleus of D1 medium spiny neurons of the nucleus accumbens, mediating genomic effects to a certain extent, at least to that initial cocaine exposure. But over time, this protein called AMP32E accumulates and begins to remove H2AZ, not only returning it back to control conditions, but nearly completely depleting this histone modification from nucleosomes within D1 medium spiny neurons of nucleus accumbens. And we have direct data now supporting the idea that this depletion of H2AZ is mediating the enhanced behavioral effects of cocaine, and our hypothesis is that our RNA-seq data will show that it's mediating this gene priming effect as well. In the interest of time, I'm going to skip over the last few slides that I had here, talking about something that was different, to end with some concluding remarks. I've been focusing at an extremely reductionistic level of analysis for a syndrome like drug addiction, which we know is mediated at the level of the brain and psychology and social cultural affairs. So why is it that we're focusing not only on a particular region of the brain, but within that region, within the nuclei of cells, nerve cells that exist in that region? Our view is twofold. The first is that providing the kind of detailed mapping, all based on unbiased data that I've emphasized to you today, we can obtain a complete map, a template, a periodic table, so to speak, of all the genes and their encoded proteins that are affected in the drug-addicted state, of the tens of thousands of proteins that are expressed in the nucleus accumbens, in brain. These data can tell us which proteins, which genes are permanently modified by chromatin scars, focusing on the receptors and transporters and ion channels located at the synapse that are being modified by drug exposure in this manner. We believe that this information will provide a robust and open-ended analysis of drug targets that can be pursued in future investigations. So with that, let me just conclude in a few ways. First, I hope I've convinced you that that chronic administration to a drug of abuse like cocaine causes dramatic reorganization of gene transcription within the nucleus accumbens. Looking at the degree to which individual genes are primed or desensitized by chronic cocaine administration in long-lived ways that last at least 30 days after withdrawal is quite compelling. I want to emphasize that that my talk today was rather narrow in focusing on cocaine and nucleus accumbens. I mentioned at the outset that the nucleus accumbens is just one of many brain areas that comprise the brain's reward circuitry, and we need to look throughout that circuitry for individual genes and proteins that are important in mediating the corruption of reward mechanisms that characterize drug addiction. Similarly, I focus today on D1 and D2 type medium spiny neurons within the nucleus accumbens. Other brain regions have other types of neurons, and all brain regions have non-neuronal cell types. And we know that drugs regulate gene transcription in non-neuronal cell types like astrocytes, oligodendrocytes, microglia, and endothelial cells, and so on, so that one needs to look in these other cell types as well, but again, on a cell type specific manner, because what happens in one cell type is very different from what happens in other cell types. And of course, we need to move beyond cocaine, as just one example, to opioids, but to many other drugs of abuse. There will be already, as from our data sets that are currently extant, many examples where transcriptional and epigenetic mechanisms are shared across drug exposure, but many other examples where they're specific to each type of drug. We view chromatin scars, epigenetic mechanisms, as one of the key mediators by which chronic exposure to a drug of abuse produces near permanent changes in brain function and behavior. Our ability to identify chromatin scars, I focused on H2AZ as one example, through open-ended proteomic analysis, I think is bearing very important fruit. Our idea is that these chromatin scars acting in concert with drug-regulated transcription factors that I've already mentioned to you today define which genes are primed or desensitized through a history of drug self-administration and drive these very long-lasting, or help drive these very long-lasting drug-addicted states. And then finally, as I mentioned recently, is that our view is that all of these data together can provide a truly unprecedented, comprehensive template for the development of better treatments for addiction. I think one reason why efforts to improve treatments for addiction over my career's time, 30 years or so, have largely failed, been largely disappointing, is because we've been guessing at which proteins to look at and not following the biology and allowing the biology to teach us which are the most important. And it is expected that what will come out of these assays are both pan-addiction treatments, mechanisms, for example, that opioids, cocaine, and other drugs of abuse have in common. Delta phosph is an example of one of those pan-addiction mechanisms, as well as many treatments that are specific to each drug type. I want to thank the National Institute on Drug Abuse, who has provided robust support for my lab over the past 30 plus years. And I do want to end with a slide of my hometown. This is an aerial view of Manhattan. Actually, it was taken by a graduate student of mine flying out of LaGuardia Airport, which is somewhere over here off slide. I don't know how the plane got over Manhattan Island post 9/11, not supposed to happen. In any event, it's a great aerial view of Central Park. This is Mount Sinai, located on the Upper East Side. And what I love about this slide is that this is the Metropolitan Museum of Art, and you can see this orange colored building, which is where I live. So thank you again very much for having me visit you virtually today, and I'd be happy to answer any questions you might have. Thank you very much.
Thank you so much, Dr. Nestler. That was a very captivating talk, and I'm so thrilled to see the future directions of your research for lifelong brain health, which is our shared mission. Thank you again so much. We do have time for questions, and I already have a few of them pop up, so I'll just ask some of them. There is a question here about the chromatin and DNA scars, definitely very compelling. Do you think that they are passed down to future generations?
So that's a great question. And it refers to the idea that the word epigenetics is used in many different ways. So I was using the word epigenetic to refer to what's happening only in a single individual, but epigenetics is also used to describe the phenomenon where a life experience can change chromatin structure in sperm or egg cells, which might be passed down to further generations. What I was talking about today only referred to what happens in the brain. Those changes are not passed down to future generations, not through epigenetics, only behaviorally. Parents with those changes in their brains might behave differently, and that might affect offspring. But in order to identify what the questioner is looking at, we would need to examine the same types of endpoints in sperm or egg cells, which several labs are doing. So it's an interesting area of investigation.
Yes, indeed. Very significant. Another question here is, do you think other behavioral addictions converge or share the exact same mechanism? You mentioned pan addiction in your end slide, for instance. Could you speak more about that?
Yes, absolutely. So, I hope I was fair in the way I described our mouse models of cocaine addiction, which I view as an inference, right? How do we know if a mouse is addicted or not? It's not the same as human addiction. When one moves to non-drug addictions like pathological gambling or pathological shopping, so on, you could imagine that studying that in a rodent becomes extremely difficult, right? Mice don't shop at Bloomingdale's, for example. The ability to understand the underlying neurobiology of those non-drug addictions is much more limited compared to drug addictions, which do have animal models. Nevertheless, there are some rodent models of gambling-like behavior, and brain imaging provides a window into human brain function. And based on those data, one can say that there is at least some similarity between non-drug addictions, behavioral addictions, to drug addiction, that the same reward circuitry seems to be corrupted in the same type of manner. So we believe that the factors that a subset of the factors that we're studying in drug addiction will be relevant for these other pathological behaviors as well.
So following up on that, cocaine is a great model because we've obviously seen that it directly impacts these D1 receptors. Other substances of abuse like cannabis targets cannabinoid receptors. Would you say that the dopamine system being the final common pathway might also then apply to other substances of abuse and could be moderated in the same way as you've described in your studies?
Yes, absolutely. So, dopamine is one signal in the brain that seems to be shared across all rewarding substances, all rewarding behaviors. So the downstream effects of dopamine would be an example of things that are shared across that spectrum. But we know that while cocaine, amphetamine, methamphetamine related stimulants work primarily solely through dopamine initially, all other drugs of abuse act on dopamine as well as other endogenous mechanisms like endogenous opioid mechanisms, endogenous cannabinoid mechanisms, GABA receptors, and so on. And so it's like the other drugs are likely to be more complicated than the stimulant drugs of abuse.
Thank you. Interesting question here. Are there currently any methods available that could silence those genes that might be responsible for addiction. So you've shown that we can moderate them in some way, but can we silence them before an addiction or the epigenetics takes place?
Yeah, absolutely. So I didn't highlight that in my talk, but it's a great point. A lot of epigenetic mechanisms are now being targeted for cancer treatment. So if you recall, in the proteomic analysis, I referred to a different histone modification, H3K79 methylation, whatever that means. And it turns out that the methyltransferase enzyme called DOT1L that catalyzes that methylation event is a major target for certain blood-borne cancers. And so there are small molecule inhibitors of DOT1L that are now in human trials for cancer treatment, which would make a trial in addiction very straightforward. The molecules are relatively safe as cancer chemotherapeutic agents go. DOT1L inhibitors are well tolerated. And so that raises the possibility that one can target these epigenetic modifications directly. So with a DOT1L inhibitor, basically undoing a type of chromatin scar and testing what effect that would have. We would still need to do that experiment first in animal models and then move on to a human trial. But I think that it is feasible and we will see it in the next few years.
Very exciting. So this is sort of like a vaccine for cocaine use disorder, would you say?
Yeah, it could be seen as something similar. You know, that to give a vaccine or a preventive agent, we know from our experience with COVID, we can only do that with molecules, vaccines that are extremely safe. And I think it's unlikely, I think it's less likely that a molecule that targets epigenetics will be safe for a lifetime, or at least it will be hard to demonstrate that it will be safe for a lifetime. So I would see these chromatin scar mechanisms as being better for treatment as opposed for prevention.
Great, thank you. Another question here is, with this information, what changes could we be making in our approach to assist with those who are suffering from addiction?
This question comes up a lot, and I've been very honest over the years in saying that when I started my lab over 30 years ago, we thought that the application of modern neuroscience and molecular biology to these questions would result in improvements much more quickly than we've seen. Arguably, the treatment of drug addiction is not so different today compared to the way it was when I started out in my career 30 years or so ago. On the other hand, I say that to be honest, but I don't say it to be nihilistic or pessimistic, because when one looks at the progress made in cancer, I think it provides a useful comparison. Cancer is much simpler than addiction. Cancer involves essentially one cell type. If one can cut out a tumor, pretty much every cell in the tumor is identical to at least a first approximation. All one needs to do is to develop molecules to end the uncontrolled cell division that's occurring in that one cell type. I was taught in medical school 40 years ago that the pathway to do that was through a personalized approach, cutting out the tumor, identifying molecular drivers of uncontrolled cell growth, coming up with an inhibitor and giving it back to the patient. Well, 40 years later, 4 decades later, we're finally beginning to see those advances. And we've seen cancer deaths decrease over the last several decades as a result. But cancer remains one of the leading causes of death, meaning that there's still a long way to go. Addiction is just going to be more complicated, like other psychiatric syndromes. And I'm as optimistic as I was 30 years ago. It's just going to take longer time. If we stop doing basic research today, we would not get those new treatments. if we stopped the research, basic research in cell growth 30, 40 years ago that I mentioned for cancer, we wouldn't be seeing the advances that we're beginning to see now. This fundamental research is absolutely required. Now, in the meantime, we need to do better in treating people with addictive disorders today. That means providing better insurance coverages for treatments that we know offer patients benefit, optimizing behavioral therapies, some of which can work quite well in some patients, whether it's AA or NA type approaches, cognitive behavioral therapy approaches, or other behavioral paradigms, feedback, brain imaging based feedback mechanisms that you've been working on, Francesca, and many other types of approaches should be looked at because they can be applied to patients today. But we can't stop the basic research because then we're consigning people with an addiction problem forevermore not to have fundamental treatments that we need.
I couldn't agree with you more, Dr. Nestler. Thank you. Another question here, are these changes permanent or could they repair themselves over time?
It's a great question. The longest we've looked is 30 days, where they seem to even build during that withdrawal period. We're now very interested in looking at longer withdrawal periods in animals. My expectation is that few things are truly permanent. And even if they are permanent naturally, there are ways to make them unpermanent, to reverse them with medications. So we need to find out exactly how long lasting they are. And if they prove to be long lasting, to think of ways like the DOT1L inhibitors I mentioned, in which we can reverse it.
Great. And I think we only have one. We only have time for one last question, so I'll end with this one. Given the significance of how genes affect behavior, could you elaborate on how this addiction tendency might be inherited from our parents?
Sure, absolutely. So I didn't talk about this, but there's actually a robust data now to show us that addiction is a highly heritable condition, that roughly 50% or so of the risk for addiction is heritable, is genetic in our DNA sequence. That's actually a really high number for a syndrome that we know is highly influenced by psychology and social cultural affairs. 50% heritable is almost where high cholesterol is and where high blood pressure is. So the fact that addiction is 50% heritable is quite notable. The other 50% is mostly non-heritable or environmental. So we know that people who've experienced adverse life events, subjected to different forms of stress throughout life, have greater risk for an addiction syndrome, and the interplay between environment and genes, the mechanisms by which environment interacts with the genome to actually determine an individual's risk can be seen as epigenetic, right? So that combination is epigenetic, determining each individual's risk.
Yep. Very good. Well, thank you again so much, Dr. Nestler, for answering all these questions and inspiring us to continue working towards brain health. Thank you.
Thank you, Jessica.
You're welcome. The power of research truly lies in how we use it to make the world a better place. We've launched a large-scale landmark study called the Brain Health Project, which will help people everywhere get proactive about maintaining and improving their brain's health and performance. If you're interested in learning more about the Brain Health Project and possibly participating in it, please scan this QR code with your phone on the screen or visit thebrainhealthproject.org. The power of research truly lies in how we use it to make a better to make the world a better place, as Dr. Nestler so very eloquently described. So thank you so much for joining us. I hope that you'll all be able to join us on May 21st for our next Frontiers talk with Dr. Ed Boyden from MIT. Thanks everyone for joining. Thanks again, Dr. Nestler.
Thanks, Francesca. Great to see you. All the best.
Bye-bye. Thank you.
Bye-bye.
We work, we live, we innovate and create. At the center of it all is your brain health. The ability to solve problems, think analytically, share empathy, and thrive.
We're trying to make brain performance really the next fitness revolution.
So how do you boost brain power? Welcome to the Brain Health Project, an urgent call to transform your mind to work stronger and faster.
This is an absolute crisis as great as any we have ever faced. We have to equip the minds and brains of our citizens to cope with the accelerating, dizzying rate of change that they face in their lives.
Your brain health is not fixed. Scientific discoveries prove it can adapt and grow regardless of your starting point.
Our greatest value, the asset that will help us to change everything, every problem we're in, is all in our head.
To harness that treasure, we must measure and monitor progress while things are going well versus waiting for an injury or disease to strike.
Too many of us are outliving our brains, and that does not have to be the case.
The information age is bombarding us with more content than our human brains can handle. How do you keep from getting lost in this? And focus on deep thinking. For starters, stop multitasking.
Science shows us that multitasking is bad for your brain. It reduces fluid intelligence, causes brain atrophy, and increases chronic stress.
The global pandemic is creating more stress than ever. Stress that leads to depression and anxiety and beyond. Unlocking our potential to navigate these hurdles starts with learning the right strategies, even in school.
So when teachers have these strategies, they're empowered to support our learners, and then the learners are now able to take ownership of their learning.
Training kids how to think is doubling academic achievement among middle schoolers.
I think the greatest national security threat is pre-K through 12. If we don't take care of educating our young men and women, then we have to ask ourselves, where are we going to be in 20 years?
Our world-renowned scientists know you can increase your brain health, not lose it. It's time for a new category of health, brain health. You are a game changer. Ready to transform the world with us? Be a part of the brain health revolution. It starts